So in this video, we're going to be thinking about membrane transport processes, so how molecules get in and out of cells. And to do this, I'm going to use an example of a marine fish, and particularly what's happening in the gills. Okay, so here's my fish. Apologies for the dubious cartoon. Okay, here's a fish. Okay, and it's living in the marine environment, which is obviously a high salt environment. Okay, so square brackets indicates concentration. That's a very common biological shorthand. And of course, salt is a mixture of sodium ions and chloride ions. So sodium ions are positively charged, chloride ions are negatively charged. Okay, so this fish is swimming about in a high salt environment. So inside the fish will be relatively low salt. Okay. So the tendency is, if you've got a high salt environment on one side, a low salt on the other side, the system will try to balance itself out. What's naturally favourable is to have the same concentration on either side. And the way to balance out concentration often is to have the movement of water. So if we lose water from the fish and put it in the marine environment, that should reduce the concentration of the salt and even the system out. That's what sort of thermodynamically is going to be happening. So of course that can never happen. The amount of salt, the amount of water in the fish is never going to balance the amount of salt in the sea. But energetically, that's what's happening all the time, is our fish is losing lots of water into the marine environment because of this high salt concentration. So our fish is losing water all the time. And of course, losing water leads to dehydration. So even though it's in the sea, it's in an aquatic environment, our fish is in danger of dehydrating because it's always losing water to balance out this salt issue. So of course, the fish takes in water and it drinks it. So it takes in water to balance that out. So you're like, okay, that's fine. But of course, that is salty water. It's taking in some of that sodium chloride. So there's sodium and chloride ions. So those are also going in to the fish. So now we've got the problem that we've got sodium in particular is the problem. So sodium is toxic. Cells can't accumulate very much sodium inside them before it starts to go wrong. So what the fish also needs to do is to excrete the sodium. So all of this salty water that it's drinking in, it needs to get rid of the salt and particularly those sodium ions. So it needs to have a process of excretion, and that happens at the gills primarily. Happens in other places as well, but mostly it's about the gills. So we've got a system where we need to be working at how on earth we can get salt or sodium ions across the membranes at the gills. So let's have a think about gills. So gills, so I'm going to draw this as a membrane all the way across my piece of paper. If you're not sure about how membranes are structured, there's another video on membrane structure and function. So membranes are made of lipids, so we've got phospholipids here. So this is the membrane. This is the external environment, and this is the cytosol. So this is on the membrane of the gills of the fish. Okay, and just as a reminder, so the external environment has a high salt content. Okay, so this is the seawater, and this is what's coming from inside the fish. Okay. Now you might be thinking, hang on, aren't gills to do with gas exchange and respiration? Well, yes, they are. That's their most important job. is to take in oxygen, which is needed for respiration. And as a result of respiration, we of course get carbon dioxide, and that needs to leave the gills. Okay, so we have these two gas molecules, and these gas molecules are able to diffuse directly across the membrane. If you remember from the other videos, the membrane internally is hydrophobic. The inside of the membrane is hydrophobic. It's a lipid-based environment. It's only non-polar molecules that are able to get through this layer of hydrophobicness. But that's okay, because oxygen and CO2 are small non-polar molecules, so they can get through the membrane by just simple diffusion. They don't need anything specialist in order to get across the membrane. They're small molecules, they're non-polar, so they quite like this hydrophobic environment. So oxygen and CO2 can just move directly across the membrane. We need to think about water moving across the membrane a little bit. So water is also quite a small molecule, and it can just about get across the membrane by diffusion. It's a small polar molecule, so it doesn't really like going through this lipid environment. Small amounts of it can, so we can have very slow, simple diffusion. But that simple diffusion is probably not going to be fast enough to be biologically useful. So what biological systems do is, if we have a molecule that can't get through the membrane very easily, is we introduce a protein. So membranes are full of different types of proteins. There are enzymes, there are receptors, and there are transport proteins. So for water, most water doesn't just pass through the membrane as simple diffusion. It goes through a transporter protein, and this is what's known as facilitated diffusion, i.e. via a protein. And that will be faster, or allow for greater volumes to go through. So if we've got a biological transporter protein in there, then we can have much more movement. And that protein is known as an aquaporin. Our transporter proteins for water, okay? Don't require any energy. It's just providing a hole in the membrane. It's a pore, so the water can go through at a faster rate through this facilitated diffusion route than the slow, simple diffusion. Okay, so that's how we usually think about gills. But let's come back to our problem of this sodium, this toxic stuff. So sodium, we know, is an ion. Okay, so sodium, here's our sodium ions. And we've got some sodium both sides of the membrane. It's an ion. It has a formal positive charge. That means it cannot get through the membrane by itself. This is too polar to get through the membrane. The membrane is hydrophobic, whereas this is a polar ionic system. So this positive charge means it cannot go through the membrane. It's basically going to bounce off the membrane. It can't go through. That's not a route for it. Okay, so we're definitely going to need some sort of transport protein. You cannot have diffusion of an ion across a membrane. Okay, so we need some sort of transporter. That's fine. So let's draw a transporter in there. So, okay, well, sodium can go through the transporter. Well, there's another problem with the transport of our sodium in this case. So, if you remember, we've got high concentrations of sodium ions on the outside, because that's the seawater, and we've got a much lower concentration in the cytosol. It's still too high, it's still toxic. We need to get that concentration down even lower, okay? But we've got a problem in the directionality of this system, because the natural tendency, as I say, we want to balance the concentration out. So naturally, what would be favourable would be for the sodium to move inside. That would be the energetically favourable thing to do. But we're trying to do the complete opposite of that. We're trying to get the sodium out, which is unfavourable. Okay, it requires energy to get something from a low concentration into a high concentration. In the downwards direction, it's favourable, that's okay. But this upwards direction basically can't happen. So that can't happen. So if we're going to get sodium across the membrane, it's going to have to go through a transporter because it can't just diffuse across. And that transporter is going to need to get some energy from somewhere. Energetically, that can't just happen in a simple way. So that transporter needs to have some cellular energy, which of course is in the format of ATP. So there's another video all about the energetics of ATP. But when we convert ATP to ADP plus phosphate, that releases energy, which we can use to do something unfavourable. So what we have in this transporter protein, we hydrolyse the ATP, and the energy of that is used to move the sodium from the low concentration to the high concentration, so it's used to do the unfavourable thing. So without that energy, we couldn't move the sodium. Okay, so this is one of the uses of cellular energy, which of course comes from respiration. The whole point of respiration is to make ATP. Okay? So that would work hypothetically, but actually biologically, that's not quite how this protein works. So what the protein does, it doesn't just move one sodium, it moves 3 sodium out, but at the same time, it also moves 2 potassium ions in. Okay, so it doesn't just pump sodium, it pumps sodium out and potassium in. So this transporter is known as the sodium potassium ATPase. Okay, so it's moving sodium and potassium, it's using up the energy from ATP, so that's its name. And this is an example of a primary active transporter. Okay, it's a primary active transporter, so it's active in this case equals using energy. So it has to get its energy from somewhere else, and in this case that's ATP. So active transport uses energy. All of this over here was passive transport. We didn't need any additional energy for it. It just happened either with or without a protein, but there was no consumption of ATP. This one over here is an active transporter, and it's called a primary one because it uses ATP directly. There are some proteins that use ATP indirectly, and there'll be another video to explain those, but this is a primary active transporter. It uses ATP directly to transport the molecule, the 3 sodium and the 2 potassium across the membrane. So you'll have noticed that that gives us a bit of a charge imbalance. So we've got 3 positive things have left the cell and 2 positive things have come out. So that means that the outside of the cell is now more positive than the inside of the cell, because there's been an imbalance in the ions that have moved across. And that difference in charge is something called a membrane potential. And there are other proteins that respond to that difference in charge, for example. So there are some what we call voltage-gated ion channels that will change whether they're open or closed depending on that charge. So we've got our primary ATPase, it's doing primary active transport, and as a result of that, it's used up the ATP, it's moved 3 sodiums out, 2 sodiums in, and we've generated a difference in charge across the membrane. So our fish is now happier, we've got rid of the sodium, that's gone out into the environment, it's managed to regulate its water, and we've used a whole bunch of different membrane proteins to do that.